Chapter 7, DNA structure and replication. Okay, so the learning objectives for this chapter are basically all centered around DNA. So you're going to be able to define the structure of DNA and how it is organized, learn how DNA is copied and amplified, understand the concept of polymerase chain reaction, and then in your lab, just still being a little developed, you guys are going to be able to understand how DNA profiling uses genetic variation in DNA sequences or their short tandem repeats and biotechnology like gel electrophoresis to help forensic scientists. So lab will likely be centered around this, but your chapter in the book is also very much centered around crime scene and DNA profiling. Okay, so let's start off. What is DNA? We all have heard DNA. All of our cells have DNA, whether it's a prokaryotic or a eukaryotic cell. All cells have DNA. Not all cells have a nucleus, so the DNA might be free floating in a prokaryotic cell where it's in the nucleus in a eukaryotic cell, but everything has DNA. So DNA is short for deoxyribonucleic acid, and it's basically your hereditary molecule. It's your instructions, right? And it's passed from the parents to the offspring, again, common to all living organisms. And it serves as the instruction manual for how to build an individual. Everyone has individual DNA. No one has the same DNA except for identical twins. Identical twins do have identical DNA. But other than that, people do have different DNA, which is why we're able to use it to profile and use it as an effective tool in crime scenes. But DNA is basically the instruction manual for how you build that individual, how each protein is built, how your body responds to different things. All of that is within your DNA. So again, DNA is found in the nucleus of eukaryotic cells. And when we're focusing on DNA for the most part, I think for most of this unit, with few exceptions, we are talking quite a bit about eukaryotic cells just because we will have a lot of like human applications to it. So when we're talking about eukaryotic cells, they obviously have our membrane-bound organelles, including a nucleus. So our DNA is in the nucleus of eukaryotic cells, and it comes in the form of a chromosome. I'm sure you guys have heard of chromosomes, and if you ever see me draw them out, I basically draw them as this little X. Hopefully a little bit better than that. But a chromosome is just a single DNA molecule wrapped around proteins. For humans specifically, we have 23 pairs of chromosomes, which means that we have 46 total. So don't get that confused. We do have 46 total or 23 pairs. Okay. And I like to think of it in pairs because it's a little bit easier once we start getting into the hereditary components of genetics. In that in these 23 pairs, you get one chromosome from mom and one from dad. Okay, so one chromosome from each pair is inherited from your biological mother and one from your biological father. So when we're looking at these chromosome pairs, the 23rd over here is the one that determines sex. So again, we get one from mom, one from dad. So if you're a female, you're XX. If you're a male, you're XY. So if you're a female, you get an X from mom and an X from dad. Females only have two X's to give, and this becomes important later on in the hereditary stuff. So mom is always passing on an X, right? So it's the dad that's determining sex. Dad can either give you an X or a Y and determine whether you're a male or female. And again, each person's DNA is unique, so you're getting different combinations, and we'll talk about this a little bit later on in this unit. You're getting different combinations from mom and dad, and then they're combining to make something completely unique that hasn't been seen before. So to understand a little bit at the beginning about why your DNA is unique, let's go through the structure and the basics of DNA. Okay. So starting with the structure, you guys have heard this word. This is one of our macromolecules, nucleotides, right? So DNA is composed of nucleotides. So this whole thing here is referred to as a nucleotide. A lot of times, I think people refer to nucleotides and you think of, just the base. It is actually this whole unit. So a nucleotide consists of a sugar, which is actually the deoxyribose. So when you hear about the sugar here or you see deoxyribonucleic, deoxyribose is the sugar. So that's that red guy right there in the middle. And then you also have a phosphate and you have a base. The base, again, is sometimes what you hear about when people are referring to the different four bases that we have, but, or sorry, the base is one of four, but sometimes I see it being called nucleotide. So just be careful with that with that terminology. Um, so with these nucleotides, they always have a phosphate, they always have a sugar, deoxyribose, and one of these four bases. So the four bases are adenine, thymine, guanine, or cytosine. So ATGC. And we'll hear this even more in just a second, but these always go together. So definitely make sure that you understand them. So when these strands of nucleotides pair up and twist around, they form this double helix, which I'm sure you guys have all heard about. And when you're looking at a double helix, what you're looking at essentially, and I know it gets complicated when you're looking at it like this, but the sugars and the phosphate, so if you remember, we have our little phosphate and it's connected to our sugar, which is connected to our nucleotide base, right? So this phosphate here, and this is our sugar. The phosphate and sugar are covalently bonded. Okay, so let's see here. That's a covalent bond. Covalent bonds, if you remember, are really, really strong. And the reason why we want them really strong is we want them to hold these strands of nucleotides together. If that bond was weak, your DNA would break and would basically become non-functional. So we need those to be really strong. That's the backbone of the DNA. And then in the middle here, we have our rungs of DNA. Those are made up from the bases. Okay, so we have our phosphate, our sugar. I know this is a bad drawing. And then basically on the other side, we have the same thing, and they're just twisting. So these two are linked up. These are the bases. Those are the rungs, and it's going all the way up in this double helix. And then over here, we have the backbone. Okay, so make sure you understand that. Again, covalent bonds for the backbone because we want them nice and strong. We want our DNA to stay together, to stay functional. Whereas our bases, those internal rungs in the middle, those are actually forming hydrogen bonds. Okay. And again, hydrogen bonds, if you remember, are pretty weak. So these two strands of the double helix are held together by our base pairing, is what we refer to it as, or hydrogen bonding between the bases of each strand. And you'll see that they're a little different. So between C and G, we actually have 3 hydrogen bonds. Between A and T, we only have 2. I know that's, I'll redraw it. Okay. So A and T, we have 2. C and G, we have 3. That doesn't, for your purposes, really mean anything. Just know that they are different in how many bonds they each have. But the important thing is that they're hydrogen bonds, which means that they're really weak, which means that they can come apart, which is a good thing. We don't want our backbone right here to come apart, but we do want the rungs to come apart because that's how we go through DNA replication, which is where complementary base pairing, oops, sorry guys, where complementary base pairing is going to come into play. So please remember, we have our 4 bases, adenine, thymine, cytosine, and guanine. A always goes with T, and C always goes with G. This will get a little bit more complicated later on when we start learning about RNA because we introduce a new base. But for right now, make sure you remember A with T, C with G. I know there's probably little mnemonics to help remember this. I don't know. The one I always just think of is I just think of at. At makes sense. CG doesn't really make sense, but I think of C and G looking pretty similar to each other. So you can kind of find different ways to remember that, but A is always with T, C is always with G. Okay. So for each individual, you have a specific sequence of these nucleotides that's unique to you. So if we want to look at someone's DNA profile, if we want to figure out what your DNA is, if you leave a little bit of DNA at a crime scene, basically what we want to do is we want to be able to visualize your unique DNA sequence which means that we need a lot of your DNA. I'm not sure if any of you regularly commit crimes or are in any sort of industry where you're studying this, hopefully not the first one at least. But if you are committing a crime, ideally, you want to leave as little DNA, if any, as possible, right? You don't want to leave blood behind, you don't want to leave hair, you don't want to leave anything where they can extract DNA because DNA is like an absolute must-have in court, right? So normally when you go to a crime scene, unless the criminal is a terrible criminal, you regularly don't have very much DNA. So we have to find a way to be able to make lots of it in order to actually create a profile. And again, a profile is basically just being able to look at the person's whole genome. Okay. And we'll talk about what a genome is here in just a second too. But with DNA replication, our bodies are naturally replicating. DNA is naturally replicating in our cells pretty much all the time, not round the clock. It replicates when it needs to, and we'll get into that a little bit when we get into mutations and cancer. Um, but the cells that need to replace themselves more often or need to go through DNA synthesis will do it when they need to. So it's happening all the time in our bodies and all the time in different cells and organisms, but it's not a constant thing, okay? But it is a natural process, and it's basically just these cells making an identical copy of the DNA molecule. The way that it does this is it takes advantage of that complementary base pairing. So A with T, G with C. So you'll remember we have those hydrogen bonds. So we have 2 between A and T, 3 between C and G. And the very first thing that we have to do is we have to get this enzyme called helicase. So helicase links up with these guys. This is the very first thing that has to happen. And basically comes in, breaks all of the hydrogen bonds right up the middle of those rungs of your double helix, holding the bases together so that they can separate. So this is kind of, helicase is kind of like what's going to go through and unzip your DNA. Okay, so there's helicase unzipping the DNA. As helicase unzips it, we have another enzyme called DNA polymerase. And basically what DNA polymerase does is it comes in and it reads the DNA and it adds whatever complementary base pair that it needs to. So if it reads a T, it's going to add a A. If it reads a C, it's going to add a G, and so on down the line. So when this is happening, we call it a semi-conservative mechanism. Okay, semi-conservative meaning if you look at our original double helix. So we have our double helix, it's in blue here, right? It's kind of 2 different shades of blue, but whatever, ignore that. We have blue. This is our original double helix, okay? And what we're gonna do is helicase is gonna bind to it. It's gonna unzip it right down the middle here, and it's gonna create one blue strand over here, one blue strand over here. DNA polymerase is gonna link up to it, start complementary base pairing everything, and that's gonna make this green strand. So semi-conservative just means that you're producing 2 copies of the original DNA molecule. They are the exact same, right? They're identical. They're not changing anything unless there's a mutation, which again, we'll get on later in the semester. But each molecule consists of 1 strand from your original and 1 new strand. So the blue is the original, the green is the new strand that we created via this semi-conservative mechanism following base pairing rules or complementary base pairing rules, okay? So we're always producing identical copies, but we've got 1 strand from the original and 1 new one. Okay, so when we start talking about polymerase chain reactions, so we know that our DNA polymerase is what's reading the DNA, right? That's a natural thing that's happening. But say we find a little bit of DNA, say we find a little bit of a blood stain from a crime scene. How do we actually understand whose DNA that is? So we've got to get that blood stain. We've got to extract the DNA from the blood. And because it's a really small amount, they go through something called polymerase chain reaction or PCR. You might have heard of this before, even with coronavirus. One of the potential tests that you can have done is a PCR test. So basically, they're just getting your DNA, which I think is pretty much only available via um, saliva for COVID, but they're getting your DNA and they're basically just finding a way to replicate and amplify a specific DNA segment. So they're taking the little bit of DNA you have and making it into a lot of DNA. And the way they do this is they add your DNA from the sample, they add a bunch of nucleotides or nucleotide bases, A, T, G, and C. They add the DNA polymerase, whose job it is to actually bind and um, read the DNA so that it can add more ones, and then the primer. The primers are just short segments of DNA that basically act as like guideposts, and they flag the section to which you want the DNA polymerase to grab onto. Okay, so once the DNA polymerase binds onto the section that the primer wants it to, then it can start adding complementary base pairs to the original strand, creating more and more strands of DNA. Okay, so again, the primers are just those short segments of DNA that guide the DNA polymerase to where it wants it to go. So when you're going through PCR, the very first thing that has to happen is normally we'd have helicase, our enzyme that unzips it, but in PCR in the lab, when we're doing this, we heat the DNA. And what that does is it separates our DNA strands. And then once we start cooling the DNA again, then we can actually allow the DNA polymerase to pair those new nucleotide bases to the original template strand. Okay. So what this does again is takes a little bit of DNA and makes a lot of DNA. So DNA replication can occur many times in a very, very quick amount of time. So you'll see we go from one strand to two in the first round, and then we go from 2 to 4. By the time you get to round 30, you've made over 1 billion copies of DNA. And PCR in the lab can happen very, very quickly. So it's easy to make this amount of DNA. And again, we can do it by our semi-conservative mechanism using complementary base pairing. So we're always making identical strands. There's never, or very rarely, any errors in this. There are things that you can do in PCR if you ever find yourself, or if you have done it before, there are things you can do to mess it up. So if you only have a little bit of a DNA sample, you want to make sure that you really do it well. You can't overheat it or cool it too fast. There is a technique to it, but overall you can make lots and lots of DNA from it. Okay, so when we're looking at doing something like PCR, we're doing it to make a DNA profile, right? And it takes advantage that there's no two people with the exact same DNA unless you have an identical twin, which sometimes can also work to your advantage when you're trying to figure out a crime scene. If, say, that person is already dead, or you can't get a hold of them, but you can get a hold of their identical twin brother, whatever it might be. But ultimately, we're looking at this big visual picture of their genome. And a genome is a complete set of genetic instructions encoded in the DNA of an organism. So everyone has their own genome, they're all unique to them. But looking at an entire genome takes a lot of time and it's really expensive, which is another reason why we use PCR. So even if, say, if you've ever done 23andMe or Ancestry.com, whatever, there's a whole bunch of them now, right? And I've never actually done it. My brother has, and that was kind of enough for me to uh, look at and kind of get the gist of what our ancestry is. But now there's lots of different ones, right? You can look at your DNA in terms of what your ancestry is. You can find people that you might be related to. You can see how your body reacts to certain nutritional factors, like if you handle proteins better. And all of this is kind of I don't want to say gimmicky, but it's all definitely coming of age. So with more technology, with better DNA technology as well, and PCR, and different lab techniques, we're kind of getting into the groove of this, and we're learning more and more all the time. So there's probably going to be all sorts of crazy things you can learn from your DNA here pretty soon. It's already pretty impressive, but our genome is huge. So going through and actually sequencing your whole genome is really expensive. So even when you send in your little cheek swab to 23andMe, they take that little tiny bit, they do a bunch of PCR, they make it really big, but they're only looking at specific sections. So we figured out a lot of different things, but we still have a lot left to learn that applies to literally everything. But especially with DNA, when we're looking at DNA, we know that there's certain areas of interest. So we might be looking at your double helix, right? Pretend this is a double helix. And we know that this section right here has to do with some certain disease, or this section has, X, Y, and Z, whatever it might be. There's certain sections that we know to look for certain things, and there's a lot that we still have left to discover. But when we're doing something like DNA profiling, trying to figure out whose DNA we're looking at, we use PCR and we only amplify, say, this section right here. And the reason we amplify this section is we know that that's a section of STRs, or short tandem repeats. And short tandem repeats are basically just a section of the chromosome in which the DNA sequences are repeated. And these are non-coding regions, so this is kind of important to know. They're non-coding, so it's kind of like the nonsense part of your DNA. It exists and it's there for a reason, but it's not coding any sort of protein. So it's not super important, but it's what makes you unique and what makes you different, among other things. So if we're looking at this STR region right here, we have the same sequence, right? So we have AGCT is being repeated in this region, the one that we said that we're going to do the PCR on. But the number of times it's repeated is different for each person. So you see here we have our paternal chromosome. On the paternal chromosome, it's repeated 4 times. On the maternal chromosome, it's repeated 6. So mom and dad both have a different section of that DNA. Some people, it might be repeated 33 times. That depends. But the number of repeats that you have are different in each individual. And keep in mind, these are huge sections. Your genome is billions and billions of copies. So these are big sections that we're looking at, even if we're only looking at one tiny section. And when we're looking at these STRs, they're always in the same place along chromosomes. And again, the exact length just varies from person to person. So we can use this to figure out whose DNA we're looking at when we're trying to make a DNA profile. So let's say we find some DNA at a crime scene, and for some reason it's cheek cells and saliva. So someone left a whole bunch of spit on the ground when they committed this crime. Okay, so we collect these cells from the crime scene. We want to extract the DNA from it, so we're going to use PCR. So we're going to add the DNA sample, we're going to add our polymerase, we're going to add some primers that tell it where to bind, we're going to add some nucleotide bases, and then we're going to amplify it. So we're going to amplify certain regions that we want to look at, STR region 1, 2, and 3. We'll do PCR a bunch of times, make bunches of copies. Once we have all of those copies from our PCR, then what we're going to do is we're going to separate those STRs by something called gel electrophoresis. Okay, so something about DNA. DNA overall has a negative charge. So because it has a negative charge, when you put it in this sample, which you guys hopefully will see at some point, basically we have this like agar gel block. So think of this little gel block and you have these little wells here. And this is where we're adding our DNA. So our STRs from our saliva sample are here. So let's say we have our crime scene sample. Let's redraw this. We have our crime scene sample here, and then we have suspects 1, 2, and 3, right? Make this even bigger. Okay, so we put the crime scene sample here. We put a sample from each 1, 2, and 3, and we're putting their DNA in here. So what we're going to do now is we're going to put that gel into an electric current that basically is just running an electric current from here to here. So this side is negative, you can see here, and this side is positive. So our DNA is negatively charged, so it naturally wants to move this way. So if it's really big, if it has lots of base pairs, if the base pairs are really large, it's not going to move very far, whereas if they're really small, then they're going to move a lot further, closer to this positive side. Okay, so again, the shorter fragments are going further, the longer fragments stay near the top or the negative side. When you're running gel electrophoresis, if you guys know, negative tends to be black and positive tends to be red. So we always say to run to red. So when you're putting the DNA in or putting your gel block in, you need to make sure that you put the DNA side on the negative side so that they can run to the red side or the positive side. So now what happens is that gel electrophoresis is going to separate out the shorter and longer fragments of that DNA sample, of all those STRs that you just amplified. So these separated fragments are going to create a really specific pattern of bands. And sometimes you can see it even without fluorescence, depending on what you're using. But you can visualize it with fluorescence. Sometimes it'll glow really nice and bright for you. And each of these bands is unique to each person. So you remember we had our saliva sample here, suspect A, suspect B, and suspect C. And now all we have to do is compare the different patterns of DNA because different individuals have different DNA banding patterns. So you can see our saliva sample here, and if we go over to sample A, we've got two kind of similar green ones, right? But they're not exactly the same. Everything's a little bit off. So suspect A we can throw out, not suspect A. Suspect B has identical STR lengths, which have a big single band, okay? But it doesn't match up with our saliva sample, whereas if you look at suspect C, it's the exact same as our saliva sample. So this is something that we can do when we're DNA profiling and trying to figure out whose DNA we see at a crime scene. This does, of course, require that you have DNA samples from suspects or from people in general so that you can compare them. But this is one way that we can do it, and we normally do a gel electrophoresis in lab. So again, we're looking at multiple STRs in this, and the combined pattern of these repeats, again, is unique to a person. So you can see with our analysis and what your odds are basically. If you look at one STR region, you'll see that 1 in 5 people share that profile. So that's not great odds, right? If we only look at one STR region, we've got a 1 in 5 chance of finding the person that we're looking for, actually less than that. We've got a 1 in 5 chance of knowing that those people might share the same one. But say you look at 15, all of a sudden now your chances are 1 in several quintillion, which basically means that if you look at 15 STR regions and you find one that matches up with your sample, that's the person. There's basically no other chance it can be someone else if it matches up exactly. So obviously this gets messy and sometimes it's not super easy to do for multiple reasons, but if you're looking at multiple STR regions, which you can do, especially with PCR and amplifying certain regions of the DNA. And again, we know a lot of these regions to look for, and we're still discovering quite a few too, but we do know a lot of good ones to look for. And so if we can compare even 15, your odds are great. And a lot of times you'll compare more than 15 STR regions as well. Okay. I know that DNA can get a little bit confusing. It's probably going to start getting even more confusing. So please feel free. I will try to post a YouTube video or two so that you guys can look through that and hopefully get a little bit of a better animation. I think animations and videos are really easy to learn from. I know that my drawing is not as great as some animations are. So please feel free to look at those. Whatever I post, definitely look at that. But do a little bit of research on your own. The book also has some decent animations when you're going through Launchpad, and those are all going to be really helpful for you to understand what DNA is and how it replicates. And specifically, we're kind of focusing on the replication in kind of a crime scene sort of way, but you can think of this as anything happening with like 23andMe or anything like that too. It's all doing the exact same thing, and it's taking advantage of how DNA replicates to get information about individuals. So take a look at that, make sure you really understand it. It's going to set up the rest of these chapters within our genetics unit.